Okay, we'll understand the genetic testing, different type of genetic test, and of course we will discuss what are the weaknesses or the limitation or the strengths of these different tests that we use in lab to diagnose them. So why the genetic diagnosis is important? Because it actually for the diagnosis, and then once we know the diagnosis, then we'll go for the disease management or the disease treatment. Though for most of genetic diseases, we are limited for the treatment option. Certainly, we don't have treatment option for a lot of these genetic diseases. But what we can do is we can go for the prevention. So we can actually identify the carrier. For the autosomal recessive disorder, we can identify the risk, and then based on that, for example, now we have this thalassemia protection program from Pakistan, and the whole idea is to protect our population from thalassemia. So, and of course, to some extent, it also offers the possibility of targeted treatment or the drug trials later. And we are going to talk about these all points. Again, I have put like all the spectrum of the mutations. So do you think that one test can fit all of these spectrum or the diagnosis of these conditions? Certainly no. So we have different genetic disorders, and because of that, we have different tests that will pick these genetic conditions. So for the rare condition, we don't know any of the, we don't have any knowledge. For example, if the patient comes to you and it have a symptom that doesn't fit with any clinical condition, and even if it does, so how you are going to diagnose it? So this is quite laborious process. It used to be more laborious, but now we have certainly very successful or certain techniques that help us to identify these disorders. For example, if the patient have first symptom, is visit the primary physician, or no identification, no disease diagnosis or misdiagnosed, then again, wrong treatment. And of course, similarly, there could be these several loops of misdiagnosis, or because of course, if you are thinking that this might not be a genetic condition, so you are treating it with or the diagnosing it with something else, and then of course this will be kind of a long process. But then if the patient goes to the geneticist or the clinical geneticist, and then we identify the genetic testing and we diagnose it, and based on that can be genetic counseling and rare disease patient organization and all these things. But still, even with all these advancement and all these, the strength of diagnosis, we still cannot identify all the genetic diseases, because we are going to talk about that later. Okay, so genetic testing and the molecular diagnosis can be at different steps. For example, either we can look on the whole genome, like पूरा genome wide कर सकते हैं या targeted कर सकते हैं. अब genome wide और targeted में क्या है? For example, if you are doing karyotyping, so you are looking all the 46 chromosome. Microarray कर रहे हैं तो भी आप पूरे genome से आपने markers लेके कर रहे हैं. And then we have whole exome sequencing, and then we have whole genome sequencing. So anyone know the difference between whole exome and whole genome? Okay, so the whole genome is that the coding portion or the exons. For example, we have 25,000 genes, और उन 25,000 genes के जितने भी exons हैं, I mean, all we know that there are intron, there are exons. So exon protein को code करते हैं, intron नहीं करते. So basically, all those can be the coding subset of a genome. For example, we have 25,000 gene, and the whole coding portion of those 25,000 is whole exome. And if you look, whatever it's coding, non-coding, regulatory, promoter, enhancer, जो भी genome पूरा का पूरा 6 billion base पर अगर आप देख रहे हैं तो then it's whole genome. But then we have some specific tests where we are not looking the genome wide. So we are, for example, we have FISH analysis, we have PCR, we have probe for a specific region of the gene or the chromosome, and then we have PCR based methods, we have single gene test, and we have panels. So panel is something if we have we are looking instead of 5,000 gene, we are looking 200 gene, we are looking 50 genes. So we are going to talk about your panel aate kahan se, and so how we have the panels. So again, we have genetic disorders, a huge category, chromosomal, microdeletion, duplication, monogenic, सारों को करने के लिए तो हम किस तरह से इसको check कर सकते हैं. So first is the karyotyping, and I'm sure you are familiar with the karyotyping, and it's also called chromosomal painting. So it's a photographic arrangement of homologous pair of complete set of chromosomes, and the karyotyping is a conventional technique, and but it's still very relevant and very useful for the clinical setting. So we are still, for example, for Down syndrome and all these conditions, so we look the feature and we go for the karyotyping. So old technique, but very useful for certain disorder, for the chromosomal disorder especially. And so what it does, it count the organism all chromosomes and under the microscope. So basically it look on the number, length, and position of the chromosomes and the centromere. So it tells us, so once we have the chromosome and now we have the software, and it make these kind of pictures, karyogram, and then we know that what is so how we does that. अब इसमें हमें cells चाहिए, so either we take the blood, or if, for example, if I'm working on some cell line and I want to do the karyotype of that cell line, so I can just quickly, I'm already at the stage where the cells are in culture, so I will start from the cells. But if I'm doing the human karyotype, so I'll take the blood, and of course then again different media, different reagent. We have a lot of reagents and media. Even now ready-made prepared cheezein aa jati hain. So you put, and then you make you fix your cells at the metaphase, and then of course once you have the metaphase, you spread it and then you analyze it. So you analyze a number of metaphases and you make the karyogram out of it. So again, very commonly used karyotype for trisomy, where we have 21 additional copy of 21 chromosome. Similarly, ये हमें translocations के लिए we use for the translocation as well. So where one part of, for example, in this one, it's a translocation between 14 and 21 chromosome. So a part of chromosome is relocated or translocated to another part. So that's also something that we look in the karyotyping. But and what time, when you are a physician, what time you actually will order the karyotype? So when you think that this might be some chromosomal abnormality, especially in the gender assessment, if you are to assess the gender, and then if there is any problem in the secondary sexual growth and these kind of things, so then you also order karyotype is initial evidence to go for the genetic testing, instead of doing these all other fancy techniques that we are going to talk later. We go the karyotyping. So let me check. No, it's not the Down syndrome. It's it can cause another syndrome. And of course it's just different region. Okay, there is one thing that in Down syndrome, even like similar feature, we have like a part of chromosome 21 relocated or added, but mostly Down syndrome is considered as trisomy 21 as far as I know. But I just gave an example of karyotyping, the use of karyotyping where we can use it. So chromosomal breakage syndrome, and if there is female amenorrhea and these kind of thing, features in the females, and these are something where we go for the clinical karyotyping. And even now we go clinical karyotyping for miscarriages, a lot of recurrent pregnancy losses or miscarriages, to see if there is any linked with parental karyotype with the outcome of the fetus. Infertility, certainly we also go for the karyotype. Okay. But what are the advantages or the limitation of karyotype? Karyotype is still a gold standard for a lot of techniques, but karyotype can only see if in the good resolution can up to 5 megabases gain or loss. जो उससे छोटी आ जाएंगी, like 2 megabase की change है, 1 megabase की है, and couple of kilobases की changes है, so that is something not in the strength of karyotype to identify those. So, and the one advantage is it look on entire genome, it look on all the chromosome, individual chromosome balanced translocations को pick कर सकता है. But then of course resolution is certainly one of the thing, and now we know that there are small deletions, small chromosomal, couple of genes are deleted in, for example, there is a 100 kb region that is deleted or added. So, and the other limitation is that, of course, you require actively growing cells. So, for example, if you have an old sample, you cannot go for the karyotyping. So these are some limitation. So, and then we go for the FISH. So what is FISH? It's a fluorescence in situ hybridization, where we use the fluorescent probe to detect a certain deletions or duplications. So basically, it's the resolution is higher. It can detect from 100 kbs to 1 MB or deletion or insertions, translocation and breakpoint. But one thing that is a limitation of this is that, so again, it's not dependent on the growing cells or the culture cells. Of course, we can actually run through the DNA. And then the one limitation here is that we know it's kind of like, for example, if you are thinking that this might be this specific deletion, so you can order for that, because of course it's a probe dependent, so it will only use that certain probe for the analysis. It's a blood sample for the karyotyping. And then you you culture the cells, and then you go for the karyotyping. And it depends, like where, for example, I was using stem cell, and of course when you grow the cells in culture, it was human stem cell, and उससे ये होता है कि आप जब cells को काफी अर्से तक grow करते हैं तो उसमें भी chromosomal abnormalities आ जाते हैं, तो उसके लिए I was just using that cells. So basically you need cell. अब वो cell कहाँ से आप ले रहे हैं, आप biopsy लेके उसको भी कर सकते हैं, आप blood से भी कर सकते हैं, तो it depends on your but ultimately you need cell for the karyotyping. अच्छा, now we have like that fish में हमारे पास एक probe होता है जो कि fluorescently labeled होता है, and then it binds with the DNA, and it because of this fluorescence we can actually detect. Fish को again, it's still used for a lot of syndrome condition. For example, the Williams syndrome, it's a 7q11.23 deletion, so it's a 160 to 1 kb deletion, तो इसके लिए हम ये use करते हैं. But again, as I mentioned, that this is kind of not genome wide, पूरा genome तो हम नहीं देख रहे. तो उसके बाद हमारे पास the other alternative is chromosomal microarray. So what microarray does, it actually look your genome, for example, the patient genome against the reference genome. And microarray, there are a lot of commercially available microarrays जो कि different strength के होते हैं. और for example, we have microarray, for example, it's called SNP6 microarray. So SNP6 had 1.8 million markers. but then we have 250K, we have others. So it depends on like the type of microarray that we are using. So what it actually compared the patient genome against the reference genome. And इसमें copy number variation आपकी जो है वो आपको मिलती है, either copy number variation is you know, a normal copy जो आपकी है जीन की उससे variate हो रहा है, either it's deletion or it's insertion, so both are the copy number variation. So it have very high resolution because it can detect, आपके karyotype can detect 5 MB, your fish can detect up to 1 MB, and then your from 100 KB to 1 MB, and then your microarray can detect in KBs, in kilobases. So a part of like small, even if there is a change or deletion or insertion in a region of chromosome, तो वो भी आप detect कर सकते हैं. And then depend on the number of probes used. As I already mentioned that there are different number of probes in different commercially available microarrays. And it not only pick the you know, copy number variation. So basically structure variations जो हमारी आती हैं उनको भी pick करते हैं. Now we have like but again, because if it's not, it's small structure variation, तो शायद उसको ये उस तरह से pick नहीं कर सकता. तो उसके लिए फिर हम next generation sequencing जो है, sequencing based techniques वो use करते हैं. अच्छा, अब होता कैसे है, you take the reference DNA, आपका DNA है, and test DNA जो कि normally उनके पास होता है reference DNA, and then of course the patient DNA, और उन दोनों को जो है वो amplify करते हैं, cut करते हैं, and fluorescently label probes use होते हैं. उसमें already वो जो आपकी chip होती है microarray की, उसके ऊपर probes होते हैं, and then your DNA binds there and they tell you in this way. And there are a lot, when you get the result, you put the result in a software and it tells you that, for example, if you look here down, so it tells you that this part is deleted. And then of course, if there is an additional copy, so it will be insertion. So you actually know insertion and deletion in microarray. Chromosomal microarray हम किन चीजों के लिए करते हैं? Developmental delay, intellectual disability, cognitive impairment, dysmorphic feature, multiple congenital abnormalities, behavioral problem, autism, spectrum disorder, ADHD-like syndrome, schizophrenia, and all others. Because again, if you look at the spectrum, तो अगर आपकी कोई isolated से phenotype है, तो then your first guess is that this is monogenic. But when you, again, हमें पता है कि ये जो multiple region delete हो रहे हैं, multiple genes, so that can have broader spectrum. So if our clinical features are little bit broader than a simple rare Mendelian condition, so then we go for the microarray. Do you have any question till here? So for now, we have looked for chromosomal, and then we looked for a specifically region, the smaller region by fish, and then we went to the microarray. But then there are others that we use for now we are coming, वही आपने chromosomal disorders को इस तरह से कर लिया. Now you are going to rare condition or the Mendelian condition जिनको हमने पढ़ा था, जो कि missense indel and point mutation indel और इस तरह की mutation की वजह से होती हैं. So now we are going to look on those mutations. For okay, blood अगर हम microarray के लिए कर रहे हैं तो we just take whole blood and we take the DNA from that. Okay, so now we have in this one we have single gene, we have several individual gene. We can look on all these things one by one. Single gene, individual gene, disease focus panel, whole exome sequencing जो कि आपके 25,000 genes को करता है, और but it's still like 1% to 2% आपका जीनोम को कवर करता है, लेकिन 85% हमारी जो म्यूटेशन होती है ना वो हमारे कोडिंग रीजन में ही होती हैं, बाकी जो होती है वो हमारे नॉन कोडिंग में होती हैं. तो एंड जब वहां से कोडिंग से हमें कुछ नहीं मिलता तो फिर हम नॉन कोडिंग की तरफ जाते हैं. सो इट्स काइंड ऑफ अ स्टेप बाय स्टेप अप्रोच टू गो फॉर दिस वन. एंड वन प्रॉब्लम विद द ओके अब PCR बेस्ड कौन से होते हैं? सो जहां पे हमें बहुत क्लियर जीनोटाइप टू phenotype कोरिलेशन पता है, where we know that this is the mutation and this is for example if we have a thalassemia patient with thalassemia, again I will give the example because this is a disease where we have genotype to phenotype relation very well established. So we have a patient where we see the thalassemia or the thalassemia-like symptom, or we have like hemoglobin electrophoresis, and when we get the result, we ask them, okay, go for PCR test or for beta thalassemia gene. So again, where we know the mutation, उसके लिए हम जाते हैं. For example, DMD जो है, Duchenne muscular dystrophy, we know that again the phenotype to genotype relation is quite established, so MPL play करते हैं. So and then we know that there are some disorder where we have the triple repeat expansion. और उनके लिए भी, if again this is very, the one key point here is how well you know the phenotypic features and link to this particular gene, so then you order for that one. So an example of single gene is beta thalassemia. So beta thalassemia is a blood disorder. Beta hemoglobin oxygen carrying capacity नहीं होती, or that's why the patient have a regular blood infusion. And again, as I earlier mentioned, that this is the most common genetic disease, rare genetic condition in Pakistan. So it's an autosomal recessive disease, and around 6,000 affected births annually in Pakistan alone. And Pakistan have carrier frequency more than 6%. What is carrier frequency? कैरियर क्या होते हैं?
That contains affected gene.
हाँ, 1 copy of affected gene. तो अब जब हम ये बात कर रहे हैं कि there is a carrier frequency of 6%, it means 6 out of 100 people are carrier. So have you seen someone with thalassemia who are not cousin? Someone who have thalassemic child, but they are not related, they are not cousin. Have you seen any of these?
Yes, ma'am.
Yeah, and the reason is because our normal population is 6% carrier. So if there is a marriage between 2 carriers, so they are going to have an affected child. Yeah. So that's why even now that whole cousin marriages thing is become somehow irrelevant to this spectrum of beta thalassemia in Pakistan. So how we can prevent the beta thalassemia, and what kind of a test or the PCR based test we go? So first we take the patient and parent, we go, Pakistan mein most commonly 5 mutations hai. IBS1, 5, FSC8, 9, frame shift codon, 41, 42, and 6 base pair deletion. So these are the most common mutation in Pakistani population. So what we do first time, so we look for the patient and parent, and हम उनकी cousin, उनकी PCR based mutation identify कर लेते हैं. अगर हमारे पास, if we are not able to identify this mutation, then we go for Sanger sequencing. फिर उसको sequence करके देख सकते हैं. So once we know the mutation, and if the couple want to have another baby, and then we can, of course, we can go for the CVS sample. We take the CVS sample, chronic villi sample, and then we extract the DNA. We do the prenatal test, and of course, we tell whether the fetus is going to be affected or the disease or normal or the carrier. So this way is one way that we, and this is very common in Pakistan. I think so most of the labs are doing. I remember right now I'm not actually aware of the status, but I remember there was a lot of center from, this was Punjab Thalassemia Protection Program, and they were doing it. And I think so now, even in the clinical labs, they are doing, Aga Khan is doing this test as well. So this is one of the common, because यहाँ पे हमें पता है कि कौन से gene को देखना है. But then we have other condition where there is no one gene disease causing the same thing. For example, we have Joubert syndrome. So Joubert syndrome में क्या है? It's genetically heterogeneous. Different genes are causing one phenotype. So when you see a patient with this molar tooth-like syndrome, you read the MRI, you look, and you're like, oh, this patient have Joubert syndrome. But how you are going to diagnose genetically that syndrome, because there are many genes, so there are different ways to go for this kind of condition. And even if we go a little bit further, अच्छा आप मुझे बताएं कि आप कैसे करेंगे if you have this kind of condition, so how you are going to treat that? Oh, not treat, sorry, how you think that you are going to diagnose this one? Sequencing, yes, but किस sequencing करेंगे? Whether you are going to sequence the genes those are previously reported, or you are going to sequence the whole genome, or you are going to sequence only the coding part of the genome. So what would be your strategy?
Whole genome sequencing?
अच्छा, whole genome sequencing हम इसलिए यहाँ पे नहीं करेंगे क्योंकि whole genome sequencing जो है ना वो आपके coding, non-coding सब चीजों को करता है, and उसकी certainly मतलब if we don't find anything, so we always go for whole genome, but it's kind of a stepwise process, na? We first we have to know one no, and then we go further, and then if there is a next no, then we go further. But again, it depends really on the cost of your test, and also like now data generation is certainly not a problem in sequencing now, because of course it's very quick, and you can sequence the whole genome in a couple of days and even in a couple of hours. But now the problem is how you analyze that whole huge bulk amount of data, and for that we have certain kind of steps जो कि हम depending on how well our characterize our phenotype is, तो उसके लिए फिर हम जाते हैं, and we are going to discuss कि इस condition में हम क्या करेंगे. Okay, so and then there is another thing. पहले तो हमने one gene, one syndrome, multiple gene, and then we have some condition where we have multiple phenotypes, one disorder, or like one gene, multiple disorder. So it's like, for example, this gene TMEM67, it actually causes different spectrum of phenotype, like different multiple phenotypes. So again, for example, if you have a phenotype and so it's a both way process. So genotype going from phenotype to genotype, or when you have genotype and then you go back and check that whether your diagnosis of the phenotype was right or it's have a different syndrome, right? For example, this agar aapko ek patient milta hai and you think that this have a Bardet-Biedl syndrome, for example, or Joubert syndrome and Michael syndrome, and then you go for the test and you find this mutation, and then you're like, okay, this is in this multiple gene, so maybe my phenotype, looking again on the clinical feature of the patient, you can say like, okay, this is actually the right diagnosis of the patient. So again, very complicated thing. अच्छा, फिर हमारे पास एक और condition आती है where we have inherited rare metabolic diseases. So around 750 conditions are caused by deficient enzyme in the single enzymes. Someone have asked the questions previously. So we have more than 750 conditions, metabolic syndrome or metabolic condition. So of course, these are quite life-limiting condition, because of course, if there is no enzyme for a certain metabolism or certain breakdown of one product. So it is going to accumulate, and later, of course, we know that it is going to be lethal for the child. And so that's why we have a lot of neonatal deaths in inherited metabolic diseases. So how we actually identify these? So we actually look on the biochemical profile or the metabolites, and then of course we go for the genetic profile. And if there is early genetic diagnosis, that can certainly improve the management strategy, because of course then you can identify the gene and then you can give the either the uski diet change kar dein ya usko enzymatic supplements de dein, so then you can actually cure the disease in this way. So अब इस तरह की जो complicated condition है, तो उसके लिए हम किस तरफ जाएंगे? So now what would be our strategy? So there are 3 steps. Like the first step is targeted panel. So what is panel? For example, it's a subset. It's a small subset that is reported for when we report something, we find a new gene in one syndrome. For example, intellectual disability में हमें एक gene मिला है, या autism spectrum में मिला है, या we have in epilepsy, we report that gene. Yeah, so that's why the reporting is very important. And now this gene is in the databases and in literature, like, okay, this was found in intellectual disability and epilepsy. And then there's a group in Japan, they find the same gene, or they find another gene with a similar phenotype. So in this way, the whole set is building, right? So now we have a set that, for example, when we look on the literature and what clinical labs usually does, so they look on the literature, they find all the genes. For example, there are agar breast cancer ke genes hai, so jitne bhi reported hai, so instead of looking for the 25,000 gene, they look for that subset of gene first. So it's a similar kind of a thing. For example, if I work in AKU, and well, I tell you, I work in Aga Khan University Biological Sciences Department. So the first, when you will come to find me, if you are coming to see me, you probably will, the first thing that you will, that, okay, you will look on the Biological Sciences Department ke jitne rooms hain. You will come and see maybe in my office. If I'm not in my office, that is, maybe you see me in conference room and all these things, but in the biological sciences. So similarly, the panel is something that is kind of already reported genes for that particular phenotype. So that is gene panel. So if we have, like, for example, the last example that I gave, that in Joubert syndrome, we have multiple genes. So what would be the first strategy? Maybe you will only look for genes, those are reported for that syndrome, right? So you will go for the gene panel. The other thing is that you look for the whole exome. So you look on the coding portion of all the 20,000 genes. For example, if you don't find me in BBS department, in the biological sciences department, you might go to the faculty office, you might go to the canteen, you might go to the library. So again, now you have broadened your spectrum to identify me or to find me. So similarly, when we don't find anything in the targeted gene panel, we go for other areas, where we don't know whether the gene is or not. So अब हमने पूरा जितने भी हमारे coding portion था genome का, उसको देखना शुरू कर दिया. All genes, whether reported for this phenotype or not, we are looking for all coding portion. But when we don't find, most of the time we have a very good diagnostic yield in whole exome sequencing, but even if sometimes we don't find, but that's quite rare, when we don't find in whole exome, then we go for the whole genome. Then we look for, maybe, instead of library or ye wali cheezon mein phir pure campus mein dekhna. So this is how we go from one step to other step. For the disorders, those are not single gene disorder, and those are genetically heterogeneous disorders. Any question till here?
यस मैम, ये जो आपने हाई कवरेज और इंटरमीडिएट कवरेज और लोअर कवरेज लिखा है, इससे क्या मुराद है?
अच्छा, ये है कि हाई कवरेज होती है क्योंकि अगर आप यू आर लुकिंग ओनली एक सबसेट ऑफ जो है ना जीन आप देख रहे हैं, तो आप उसको कितनी दफा सीक्वेंस करते हैं, बिकॉज समटाइम ये होता है कि हमारा जो सीक्वेंसिंग है वो उसको आइडेंटिफाई नहीं कर पाता ना, तो हम एक कवरेज करते हैं. फॉर एग्जाम्पल वी हैव 30x, वी हैव 100x, तो हम 30 टाइम उस रीजन को सीक्वेंस करके फिर देखते हैं उन सब में उन 30 टाइम्स में कितनों में म्यूटेशन है, और वो हमें दो चीजें बताता है. अगर तो वो 30 टाइम सारे की सारी पॉइंट पे म्यूटेशन है ना, तो इट्स मीन कि ये होमोजाइगस म्यूटेशन है, मतलब दोनों अलील में म्यूटेशन है. अगर वो 30 टाइम्स में 15 टाइम्स एक में है, 15 में दूसरे, तो वो हमें हेट्रोजाइग, तो इट टेल्स अस द स्टेटस ऑफ homozygosity or heterozygosity. It also help us to identify the mutation with the highest kind of coverage. So coverage is basically like how many times you sequence that region. So if you are sequencing these 400 genes, so how many times you are going to sequence these 400 genes? So this is, and then of course similarly you can have 220,000 or 25,000 genes, and then जैसे जैसे हम बड़ा रीजन करते जाते हैं ना, तो हम उसकी कवरेज भी थोड़ी कम करते जाते हैं, because of course it's more costly, and of course it's more time consuming, and more importantly it's more कॉस्ट और टाइम कंज्यूमिंग के बाद इसका जो मेन पॉइंट है वो डाटा एनालिसिस का है. तो अगर आप बहुत हाई कवरेज के साथ करते हैं तो यू हैव a lot of data जो आपके लिए, आपको फिर उसके लिए सुपर कंप्यूटर्स चाहिए. You cannot analyze those in a normal computer and normal setting. So these are the all points. Anyone have more questions? Because this is okay. Tell me if you have ordered any of these tests for your patient, or if you have seen anyone doing this in clinical settings.
यस मैम, आई हैव सीन एक्चुअली, बट जैसे मैंने इसी तरह से बहुत ज्यादा सुना है, जैसे कोई बच्चे उनकी फिनोटिपिकल अपीयरेंस थोड़ी सी डिफरेंट है, लेकिन पेरेंट्स को इस तरह की हिस्ट्री नहीं दे पा रहे हैं कि भाई आप कोई पर्टिकुलर सिंड्रोम के साथ उसको लेबल करें, तो फिर डॉक्टर सलमान करमानी ने फिर पैनल एडवाइस किया था कि पैनल करवा लेते हैं.
Yeah. So Dr. Salman Karmani in AKU. So basically in Pakistan there are certain clinical setup where they send samples for gene panel, but so far we have to outsource it. हमारे पास वो सिंगल जीन तक तो हम कर लेते हैं थैलेसीमिया वगैरह, but we unfortunately don't have our local capacity here. But for example, Dr. Kirmani and some other people in Children's Hospital Lahore and other clinical settings, so they are sending it abroad. One thing is it's a bit cheaper, of course, क्योंकि उनके पास तो हफ्ते में hundreds of thousands of cases आ रहे होते हैं, तो because when they have workload, तो वो फिर थोड़ा cheap भी होता है. But आप जब patient, वही बात जो आपने की है ना, when you have the clinical history and you see like, okay, this is something that probably can fit in epilepsy panel. This is something that can fit in muscular dystrophy panel. And for example, now we have other, like a lot of panels are available, and वो update होते रहते हैं. जैसे जैसे नए genes add होते हैं, तो वही company जो sequencing कर रही होती है, तो then they add those genes as well. So for example, we have this human breast cancer panel, 45 genes का है, and I'm sure there are even more updated panels as well. So it include the BRCA1 and BRCA2, but of course other genes, और उसमें हमें पता चलता है अगर हमें इसमें mutation नहीं मिलती, so then we go further. But before the further, अब वही वाली बात है कि we studied that point mutation thing, कि we have point mutation, we have missense and we have nonsense. अब nonsense में तो हमें पता है कि वो pathogenic होती है mostly, लेकिन missense जो है वही बात जहाँ पे मैंने आपको, if you remember my last जो previous presentation थी, so there we have mentioned that the tyrosine is changed with the arginine. So now that arginine or tyrosine change, how that change is going to affect the protein. So अब वो जो हर change है वो तो mutation नहीं होती, तो फिर हमें पता कैसे चलता है कि what is mutation, what is मतलब what is pathogenic, what is not. So अगर हम ये देखें ना, तो ये एक spectrum है. बेसिकली कुछ म्यूटेशन होती हैं जो पैथोजेनिक होती हैं, जो आपके डिफरेंट कॉम्पिटिशनल मेथड्स जो है ना, बायोइन्फॉर्मेटिक्स और उसको यूज करते हैं, वी हैव अ लॉट ऑफ tools that we can use to check the pathogenicity or the of the mutation. So some tools they tell us that this mutation is clearly pathogenic. It have, and then of course protein interaction and a lot of in silico things that you can do. And then there are some jo ki likely pathogenic, and so these two are quite, when we have the result that this mutation is pathogenic or likely pathogenic, it gives us confidence that this is the right change in the DNA that is leading to this disease. So we diagnose it. And then we have certain, that is where we have likely benign or benign. So if we have this condition, so then again it tells us, okay, this might not be the cause of the disease. So we really don't look into that. And to do so, we actually look on a lot of computational tools. But then there is a middle spectrum that's more challenging for the clinicians. So one biggest challenge for the clinician is VUS. So VUS is something up where you can say like 50-50. So it can be benign, it can be pathogenic. Okay? Ab is case mein, when you are a clinician and you are looking on the patient report and you have this like VUS, you basically don't have, I mean, based on this result, you cannot tell patient that this is your cause of disease or this is not the cause of disease. So usko karne ke liye, I think that's a big challenge. And then we go for the functional evidence and we add that mutation into the ClinVar hai, clinical variation database or other databases for the human clinical phenotypes. And then agar kisi aur ko bhi usi phenotype mein wahi mutation milti hai, to then it give, or koi aur us pe functional evidence de deta hai, to then it gives a status of pathogenic, or agar nahi matlab koi aur functional evidence deta that this is not the mutation that is disease causing, to then the status of VUS change. But right now VUS is a very, very challenging thing for the clinicians. Okay, for example, this was one paper that I saw on relevant frequency of mutation in young women with breast cancer. So again, we know that most common cause is BRCA1 and BRCA2, and then but then there are other genes okay involved in breast cancer. एक तो इस तरह की हमें picture मिल जाती है ना, using the gene panel, and then of course it also tells us के how important the gene panel is, and जैसे जैसे नए genes आते रहते हैं, तो then we add those genes into the panel. But then there is one thing, जब इसमें से हमें कुछ भी नहीं मिलता, and certainly there are times when we don't find anything. So then what would be our next strategy? अभी हमने बात की थी gene panel negative cases. So what we will do with the gene panel negative cases? Whole exome हम पहले जाएंगे. Yes. Basically, अगर आप exome और genome को देखें ना, so this is our DNA, and then we have introns जो कि protein code नहीं करते, and then these yellow portions are exons. So exons basically have the information of proteins, of messenger RNA, and ultimately those messenger RNA is translated into protein. So if you compare the whole genome and whole exome, so basically this one slice is whole exome and the whole apple is the genome. So out of whole apple is this tiny slice that is 1 or 2% of the whole apple is the whole exome. So up and this have the coding portion of 25,000 genes or 30,000 जितने भी human genome हैं, तो so in for the exome, that is 1 to 2%, and that is our next strategy when we don't find any in the clinical settings. Research may we have different research questions. For example, if I want to look on all coding and non-coding for autism patients, so I have phenotypes and the analysis and these parts, and of course, according to that, I will order the panel.
Excuse me, sorry to interrupt.
Achha.
Participants and everybody so much involved. I think they have forgotten the break. अगर नहीं लेना चाहते, it's well and good.
अगर लेना चाहते हैं तो बताएं.
Dr. Amreen जरा पूछ लें, please.
जी बता दें. You want to take break?
मेरा
Or else
तो
you want me to?
इंटरेस्ट बहुत ज्यादा है और मैं तो यही कहूंगी कि खत्म कर लेते हैं इस टॉपिक को, उसके बाद फिर नेक्स्ट टॉपिक से पहले ब्रेक करने, बाकी जैसे पार्टिसिपेंट कह रहे हैं.
हाँ, I think so. जैसे सब कहते हैं. Okay. Everyone is saying continue. I hope you are understanding something. Okay, good. अब next हम whole genome की तरफ चले जाते हैं. अब now there are some very, very interesting things. And so one, if you know, Human Genome Project जो है, वो $2.7 billion का project था. And that was कि हमने पूरा human genome sequence करना है. And it took 15 years for the researcher from multiple countries and hundreds of labs doing that. And they were able to finish the first draft of Human Genome Project in 2001. So that time we actually sequenced those all 6 billion base pair. Uska jo order tha na, wo humne sequence kar liya. And it was 2.7 billion plus US dollar plus 15 years, hundreds of scientists, thousands of scientists, hundreds of labs. But now from that 2001 till 2020, or like basically 2021, because the figure that I have put at the end is $500, the latest one with the 30x genome coverage. So out of itne sari jo cost thi, now we have reduced the cost of whole genome sequencing, and now we can do it in $500 for whole genome and $300 for whole exome. So it's getting cheaper and faster. So previously we jahan 15 years lage the, now we can actually do in hours. So I have a very cool example later at the end of other presentation where we'll say ki kitna jaldi ab hum kar sakte hain. So there is a huge development, but again, and it's cheaper, but again, we have to look on the need. If it can be done and we can order it, but of course, do we really need it? So the current cost is $1,000 to $500. Again, 500 is for the research one, but when we know that research or clinical is a different thing. For example, if research is something jisme I'm not reporting really, because I have to go further analysis and then functional analysis and all these things, mouse model, animal model. So I have a lot of steps where I'm going and taking that analysis. But if I'm reporting the family, so it need to be in the clinical, जो उसकी quality में होना चाहिए. So trio based testing हम करते हैं. अगर हमें नहीं समझ आ रही होती तो हम parents और बच्चे दोनों का लेके कर लेते हैं, because then it gives us not only inherited one but the somatic one जो बाद में de novo mutation है. Or this is trio is something that is done in a lot of clinical setting. Ideal समझा जाता है. Rate is around 50%. Well, the phenotype success rate till 60-70% here at AKU in the clinic as well for some disorder, but for some disorder it's quite low because again depend on the clinical heterogeneity of the families and all these things. So okay, one thing that is very, very important. आपने एक जीन तो सीक्वेंस किया, उसमें तो आप we look the patient and phenotype and genotype relationship, but then there are things जो कि जब आप पूरा exome या पूरा genome कर रहे हैं तो अभी हमें पता है कि these are the mutation that increase the risk of breast cancer, for example, and you have done tests for neural phenotype, but then you find because now you are looking at the whole genome and you tell okay, this is another you have the risk. So again, there are some ethical issues, and how you communicate those incidental finding to the family, that is one thing. So this is a pipeline, how we analyze it, how we but that's very, very complicated things, but that's not a clinician job, it's a bioinformatician job. So but this is just how we do it. Next generation, these are the common gene panel that people are using, and here we at AKU we are also using. It's for epilepsy, ataxias, other a lot of other phenotypes, and breast cancer is also one of them. But then agar wahan se panel se nikalta hai, then we go for whole exome, and then we go for the whole genome. But again, the most common thing is, or the most important thing is phenotyping. So agar aapki phenotyping अच्छी है, तो then आप हो सकता है कि आप पैनल से ही आपकी identification हो जाए disease की. So I think so this is where the clinician job is most important. Okay. I think so we are quite at the end. So अब ये genetic testing, एक तो हमें disease gene identify करते हैं, उसके अलावा हम क्या, where is the future, and informed reproductive decision making. For example, if we know that this patient have this couple have a mutation that is a disease causing, or एक sibling में है और इस तरह की rare condition है, तो हम we identify the carriers if the both partner are the carrier, and then autosomal recessive की हम उसकी frequency को calculate करके उनका risk बताते हैं. Then of course we also use for the pre-conception period, you know, if a couple is going for IVF techniques and these kind of techniques, so then we go for this one. And the most common one that even in Pakistan we are doing for thalassemia is the prenatal genetic testing, जिसमें आप CVS sample लेते हैं अभी तक. लेकिन of course बाकी now there are some other techniques, जैसे non-invasive testing हम कर रहे हैं. मदर का ब्लड लेके उसमें से सेल फ्री DNA लेके हम fetus का भी चेक कर सकते हैं. तो we don't really need CVS sample, but of course here we are still using the CVS sample. So these are the things that we do. And yeah, so CVS sample. And so what are basically take-home messages? So different genetic disorder will be picked by a different genetic test. So when the patient comes, you see the phenotype, and then you go for the test. So underlying genetics of the disorder stay crucial to ordering the right test and interpret the right result correctly. For example, अगर आप एक benign mutation को ये कह रहे हैं कि हाँ ये cause है, और आप उसकी बेस पे prenatal diagnosis कर रहे हैं या carrier screening कर रहे हैं, तो it's mean like you are doing something completely off the track, na? So again, as I mentioned earlier, that data generation is not a problem, but interpretation of data is definitely now more important. And that's why, okay, so now we have huge databases in the world. There is, for example, there is this database called GenomeAD. उसके ऊपर और इस तरह से clinical variation database है. And then of course there are some guidelines for American Medical Genomics की जो guidelines है genetics की, and many others. So they tell us how to interpret the result और किस तरह से हम जो है वो interpretation करते हैं. Advantages and disadvantages both techniques के होते हैं. Of course there are some limitations for one technique, and that's why we go for the next one. Of course, but it's a stepwise process. And yeah, so genetics is continuously evolving. So for example, now we have a VUS in a patient that is variant of unknown significance, but maybe in 6 months or a year, I will know the status of that VUS, that whether it's a disease causing or not, because of course the people are putting the knowledge on these databases. Genetics is not tough, just needs to brushing of the concept in every now and then, of course, because now we know कि पहले हम inherited वाले देखते थे, but हमें पता है कि अब de novo इतना common हो गया है. And the other thing is that we just look on. So now we have like this one there's a lot of tools where those connect the clinicians. For example, if you are working in Pakistan and you have identified a mutation in one gene, and there's someone working in Australia, another person is working in USA, and they found mutation in that gene, similar phenotype. So आप एक tool है जैसे हम gene matcher कहते हैं या matchmaker कहते हैं. So basically it's I call that like a dating app of geneticists, because of course when you know the patient mutation, you put your mutation or the patient phenotype there, and if there is someone in other country and they have the same one, and then you get a message that you have a match, and then you can collaborate, and then you can actually have more strong clinical cases and strong clinical information, and that's why you can actually publish and report, because of course now you have more evidence. And unless के जब तक आप वो हो सकता है कि ऑस्ट्रेलिया वाला पर्सन क्लिनिशियन उसको 4 सालों में पब्लिश करता है या नहीं करता, and आप उसे करना चाहते हैं, but you don't get additional information. So this is one way to go through this all things. So yeah, we have epigenetic. We are not going, but I can talk about epigenetic. Actually, we will talk about epigenetic in our other complex value talk, the last one. Actually, epigenetic changes mean heritable. Yes, there are some changes in the DNA, still, not, for example, if it's like how your chromosome, chromatin open, ठीक है, जो आपकी regulatory changes हैं, तो वो आपकी जो है वो transfer होंगी. अभी हम एक technique, एक नई है, beside this sequencing, it's called ATAC-seq. तो basically वो आपका ना जो आपकी replication होती है और आपकी ट्रांसक्रिप्शन होती है, आपका डीएनए ओपन होता है. सो हाउ दोज मतलब वो चेंज आपका डीएनए अलाउ द पॉलीमरेज टू कम इन सिट एंड ट्रांसक्राइब, तो उसको कैसे अलाउ करता है, तो हम उस पूरे प्रोसेस को देख सकते हैं, पूरी उस दैट्स कॉल्ड ATAC-seq, एंड उससे हम डिफरेंट स्टेजेस पे भी देख सकते हैं. फॉर एग्जांपल वी कैन सी लाइक एम्ब्रियोनिक स्टेज पे कैसे है, अल्टीमेटली एडल्ट पे कैसे है. सो ऑल दीज चेंजेस डेफिनेटली मोस्ट ऑफ देम आर हेरिटेबल, जो कि आपके क्रोमेटिन में वगैरह में और इस तरह की चेंजेस है. बट अगेन genetics is the things that I have tried to put here was for the basic understanding, but it's very complicated and it's very kind of different than what we have read in the books previously, like autosomal and ये वाली चीजें. Because अभी एक और चीज, जैसे मैंने कहा प्रोटेक्टिव वेरिएंट है जो कि आपको डिजीज से बचाते हैं, इसी तरह से मॉडिफायर्स भी होते हैं. तो अगर एक बंदे में एक ही म्यूटेशन है, two patient have exactly the same mutation, but they have a different disease spectrum. So because एक में बहुत severe है, एक में milder है, because there might be something जो कि उसको modify कर रहा है, and we already know the functional evidence of these modifiers कि ये किस तरह से disease जो है उसको उसकी severity को increase करते हैं. So tell me if you have questions, then we will go for the break and then we'll have our last talk.
एक्चुअली मुझे ना एक थोड़ा सा वो कंफर्म करना था कि जैसे एक बहुत कॉमन सा सिनेरियो, बहुत कॉमन तो नहीं है लेकिन बट वी हैव हर्ड लाइक दैट कि वो बच्चे एक्सचेंज हो जाते हैं और उसके बाद फिर वो जेनेटिक टेस्टिंग के लिए एडवाइस होता है, फिर आइडेंटिफिकेशन होती है रियल पेरेंट्स कौन से हैं. तो जैसे नर्सरी में, चाइल्ड नर्सरी में, तो वो स्पेसिफिकली कौन सा टेस्ट होता है या क्या कंडक्ट करते हैं, एडवाइस करते हैं?
अच्छा, उसके लिए अगर आपने पेरेंट्स का करना है तो माइटोकॉन्ड्रिया वगैरह कर सकते हैं, वाई क्रोमोसोम वाला कर सकते हैं, बिकॉज आपने यू हैव द पेरेंट, और STR बेस्ड प्रोफाइलिंग जो हम फोरेंसिक में करते हैं ना, सो हमारा वो वाला होता है. सो बेसिकली हमारे ह्यूमन में बहुत सारे, तो बेसिकली तो सबका जीनोम सेम है. इट्स अ वेरी माइन्यूट changes that we have, but still we look very different, we act different, we have different kind of diseases, and but there are kuch regions jo ke bahut polymorphic hain hamare genome mein. For example, there are some regions जिनको हम as a marker use करते हैं, as a tool use करते हैं, जैसे STR markers हैं 13 regions, जो कि अभी तक फोरेंसिक्स में वही यूज होते हैं. तो बेसिकली दैट्स ऑल्सो जो कि आपका फॉर एग्जाम्पल अगर आपको क्राइम सीन से जो डीएनए मिला है एंड देन यू टेक लाइक जितने भी आपके आपको लग रहा है सस्पेक्ट्स हैं आप उनसे लेके उसको मैच करते हैं. सो सिमिलरली आप बच्चे से लेके और उसके पेरेंट्स से लेके इजी सा माइटोकॉन्ड्रियल भी कर सकते हैं, आप एसटीआर प्रोफाइलिंग भी कर सकते हैं और आप वो भी कर सकते हैं वाई क्रोमोसोम वाला कि फादर और बच्चे का अगर मिल रहा है.
is that the answer of my question which I asked initially कि mitochondrial inheritance का clinical application कहाँ होती है? So
yeah, but नो ऑफ कोर्स माइटोकॉन्ड्रिया की वैसे भी बहुत सारी क्लिनिकल एप्लीकेशन डिजीज के रेफरेंस से भी होती है ना. सो आई वाज थिंकिंग अबाउट द डिजीज रेफरेंस बिकॉज दिस इज नॉट अ कॉमन सिनेरियो कि बच्चे इतने मिस होना. सो इसीलिए वो मेरे माइंड में नहीं आया. बट यस यू आर सर्टेनली समथिंग दैट वी कैन गो फॉर द माइटोकॉन्ड्रिया. यस,
thank you.
if we have more questions. And then we have one talk. So can you tell me what time we can, like how long this break would be and when we can come back? Organizers? Okay, 15 minutes and then, okay, so we will be back. Now it's 11:30, so 11:45. And then we have one talk and then we can go probably quickly. Okay, so see you at 11:45.
Okay.
Thank you very much, everyone. So let's have 1 minute break.
Dr. Amir, we can start now.
Okay, so should we start? Okay. Okay, I can start. So I think so everyone is back. Okay. So we have in our previous lectures, we have talked about pattern of inheritance, type of genetic diseases, and then diagnosis of genetic diseases. But when we talk about the type of genetic diseases, we broadly talk about 3 types of genetic diseases: chromosomal, rare Mendelian or monogenic disorders, and then the third one was complex disorders. But when we talk about the genetic conditions diagnosis, then we mainly discussed 2 types. One was chromosomal disorders and one was monogenic disorders. So now we are going to talk about the complex conditions and polygenic risk score, and then later some environmental factors. Plus, at the end, we will try to look something on like in the future, like where is the future of this clinical genetics or clinical genomics. So let's start with the complex disorders. So the complex disorder, sometimes also called multifactorial disorders. So they are kind of not caused solely by the genetic factors, but it's called in which there are mutations. Okay, if we go back to that spectrum of the mutation and spectrum of the copy number single nucleotide polymorphism, और वो जो उसकी वजह से phenotypes होती थी, तो at the end we have this complex condition. So that is caused by multiple genes, common variation in the multiple genes, as well as and that's called polygenic, because monogenic is because of single gene, the polygenic is the mutation in different genes plus environmental factors. So that's why they are called multifactorial as well. So it's called gene and environment, and even the gene is not single gene, it's multi gene, or it's mutation in multiple genes. But those mutation are not rare mutation, those are mostly the common mutation and a cumulative effect of environment, and those common mutation actually leads to certain diseases that we call complex disorders. And complex disorders are something that are not they not follow the classical Mendelian pattern of inheritance, because of course एक तो ये है कि how many complex variant then you have, and then what is the influence of environment and how much is the influence of environment. A lot of complex condition, we have 7,000 rare Mendelian condition, but we have a lot of complex as well, where our progress in genetic diagnosis is not as high as in rare disorders. We are going to talk about that later. For example, we have heart diseases, we have hypertension, asthma, we have neuropsychiatric disorder, bipolar, schizophrenia, diabetes, cardiovascular. These all comes under the multifactorial or the complex diseases. So again, if you look at this picture, so here the genetics is, for example, in the red color and the environment is in green color. So for these like DMD, Duchenne muscular dystrophy, hemophilia, phenylketonuria, these rare condition, we have more genetics. We don't have any environmental effect. But in then there are some जो के in middle spectrum में आती हैं, and then we have environmental conditions जो के or the the बिल्कुल environment की वजह से होती हैं, pathogenic जो हैं हमारे diseases, tuberculosis और इस तरह की, जिसमें genetics का as such इतना part नहीं होता. But now we even know with this whole COVID situation, there are a lot of evidence कि कुछ population जो है वो COVID से severely affect हुई, कुछ population नहीं हुई. even with the same variant. So what was the difference? So so people have studied the host genome, like human genome, not the COVID wale genome, and then they looked that the people who was severely affected, of course, one factor was age, and then there was some genetic factor there as well. The people who had mutation in immunity related genes, so they were more they had more severe symptom of COVID-19, and of course most of them died because unki immunity already bahut compromised thi. So of course now we know that even with the purely environmental condition or the pathogenic condition, still the host genetics or the host genomics still play a part in this one. So this is the whole spectrum of how genetics and environment actually interlinked, or where the environment play roles in genetics as well. अब हमें हमारे पास genetic factor के बारे में evidence कहाँ से आती है? I mean, how we know that this is a genetic condition? For example, we mostly say like, oh हमारी फैमिली में हार्ट का मसला है या ये वाली ये जो हमारी हम कहते हैं कि ये तो हमारी खानदानी जो है बीमारी है. तो how, if for example diabetes, and it's again not one gene is not causing it, so where we get this genetic evidence? So we get it from the familial risk. So एक relatives के दरमियान में उस genetic condition का कितना risk है? ट्विंस स्टडी से हम लेते हैं मोनोजाइगोटिक ट्विंस जिनका कंप्लीट जीनोमिक पैकेज जो है वो सिमिलर है, बट स्टिल इफ द डिजीज दे आर हैविंग इज not similar, so then it might be that something is coming from the environment. And similarly with the dizygote twins, adoption studies bhi karte hain, because of course now the genetics is completely different, and at least if the adoption is from the family, so it might have some genetic factor, but again the environment ke wo kis tarah se role play kar raha hai. So these all things. Okay, so and then we have population or the migration studies. Again, the population studies also tell us that what is the risk in the population, and also migration se hame environment ka pata chalta hai. So these all factors actually take us to find ki kitni genetic evidence hai aur kitni environmental evidence hai. So evidence from these type of study can estimate the heritability of the condition, the proportion of specific genetic factors, and environment ke kitna ho sakta hai uska role. So these kind of studies is it something related to my microphone? Probably it's not.
No, I
Right?
asked them to check at their end because we
Okay.
are having a problem. Thank you.
Okay. Then we have okay. Lekin we are dependent on statistical analysis to identify the risk of this heritability and these things. So the genetic risks are not directly measured. तो ऐसा नहीं है कि आपने वो एक ही mutation आपको मिल गई and then you looked on the patient, you looked on the offspring and you identify the risk. For example, in the rare condition the way we do, but of course here we have overall हम एक population को देखते हैं and उस पे फिर हम different statistical analysis करते हैं and then we come up with the with the conclusion that how much heritability it have. So of course it's genetics and statistics is quite important in this case as well. Okay. Then अब हम करते कैसे हैं? For example, आपने familial risk करना है, so you have probands, you have the patient, and then you have siblings. और आप फिर देखते हैं कि how many of the siblings have a risk of the same condition, or having the कितनों में वही condition है, ठीक है? तो this is how, like for example, you have probands and then you have 4 out of 100 siblings, and then you can calculate the probability of reoccurrence, that is 4 out of 100, and this means 4% have the familial risk or the sibling risk. And similarly, अब हम population में कैसे देखते हैं? तो we take the normal healthy population and we take the disease population same, तो फिर उसमें हम देखते हैं कि कितनों में reoccurrence या इसका risk है, कितने further affected हो सकते हैं इस तरह से. तो this is the way that we calculate the risk. Similarly, I put this example of schizophrenia here. Schizophrenia is very challenging neuropsychiatric condition, a lot of genetic evidence, but again we could not pinpoint कि cause क्या कर रहे हैं ना. हमारे पास बहुत सारे rare variant भी हैं, common variant भी हैं, environmental factors भी हैं, and a lot of other conditions associated, and we know that the socio-economic factors and all these things, environmental unemployment and these kind of things also increase the risk of schizophrenia. But when it comes to genetic risk, for example, in identical twins, it have 48% risk of reoccurrence or the heritability. It's mean they have 100% similar genetic, but still the disease is probably, there are factors then beside the genetic factor, or maybe something with the regulation and how the environment is controlling the regulation of the gene, or how the environment is controlling the function of the gene. So this might be an important factor. So again, siblings mein kitna hai, parents mein, and certainly we know that general population mein 1% risk hai, but then we know that if you have a first cousin with the schizophrenia, so you already have a double risk than the normal population, and you have uncle or aunt with the schizophrenia, you again have double risk than the normal population. So this is the thing. So अब हम इसको दो तरह से study करते हैं इस risk को. So one is we go through the linkage analysis, we go through the familial studies and see कि क्या उनका क्या status है. Another is that we take association analysis. An association is you take a patient group and you have a control group. So it's basically case control association study. So हम इसके through जो है वो identify करते हैं. Mostly these are the two types of study. For example, if you have a patient with schizophrenia and कि उनकी family में भी run कर रहा है, तो you take the whole family and you look the genomic picture, and even अगर if you go for whole exome or whole genome sequencing, you look के कौन से variant आ रहे हैं, whether it's common, whether it's rare, and if not, तो then you take family, patient and control. So patient and control is quite a prominent model in most of complex disorder, and so basically patient in control दो तरह के model होते हैं. So one is called genome wide association study, and one is called then there is a second one. So genome wide में क्या होता है? You don't know anything about genetics, and you are not, जैसे हम पहले वो single वाले cases में हमने किया ना, that there was a subset reported, there is this thing reported. No, in this case, we are not thinking about anything. We are just blindly looking on patient and control. So our patient or control को हम कर क्या रहे हैं? We take the patient set and we take the control set, and we look for the पूरा genome में हम microarray base test करते हैं. So we look for all this common variant जो कि already population में reported है, more than 1% of the frequency, and then we look on all those. So basically, we don't have any biological hypothesis in this one, because we are not thinking कि इसमें ये gene होगा या वो gene होगा. We are just looking on the whole genomic picture, and no genomic location, because we are again looking for the whole genomic region. So if we get the knowledge of common genetic variation and large scale genotyping. So one thing that is very, very important for either it's genome wide association study or normal genetic association study, the one thing that is very important is the sample size, because we are looking for common variant जो कि already population में present है, and अगर हम उन common variant को we are going to link it with the disease, so we the sample, the more bigger sample size, the more statistical power we have. So for example, this was a study that was published in 2014 in Nature Genetics, I think so, Nature. So what they did, they actually took 40,000 cases and around 70,000 control, 40,000 schizophrenia patient and 70,000 control from mostly from European ancestry. And they looked for whole genome. Like for example, if you look down, these are chromosome 1, 2, X and Y. So all the autosomes and all the chromosomes. And then this is the significance or the p-value, like how kind of, significant the result was between the cases and the control. अच्छा, so now ये जो आपको ऊपर नजर आ रहे हैं, it's called Manhattan plot. So इस तरह का जो ग्राफ होता है, if you see this red line, so this is basically threshold. अब this is our statistical threshold. So whatever is above this is significant. And we know that all small points, of course, you cannot see here because it's a millions of SNPs. So हमने पूरी million of SNPs को in 40,000 cases and 70,000 control में देखा. And what we have seen at the end, that there are certain regions, there was actually in fact 108 regions that were common, that were more significantly associated with schizophrenia as compared to control group. And the most common or the biggest association was on chromosome 6, MCH major histocompatible complex. We know that there's a lot of evidence with the schizophrenia coming from immune system. We really don't know, because जब तक हमें functionally इसका नहीं पता कि किस तरह से ये कर रहा है, but we know that there is some association even in this thousands of samples. The other common genes that the SNPs that we have found was on DRD2, dopamine receptor, GRM3, glutamate receptor 3, or ORB, because we know that even in schizophrenia, there is the dopamine, there is the glutamate hypothesis. So this kind of study, we go for whole genome sequencing, whole genome, genome-wide association study. The other one that we mostly do in Pakistan is association study. So basically, they took thousands of samples and millions of SNP, and they look across the genome. So now, what we do usually, we take some SNP. For example, अगर हम इसकी ये SNP जो है, जो कि most significantly associated है, या ये वाली जो कुछ SNPs हैं, जो कि most significantly associated हैं with schizophrenia, we only take those SNPs, 2, 3, 5, whatever number is convenient in our research setting. So then we take our, we don't have any patient uncontrolled genome-wide association study. I'm sorry for this. I think so there's like some problem because my control OL is here. But one is case and the other left side is the control. So basically we take उनके जो result में significant आए हैं, and we only look for those SNP in our population, and that's what we are doing for most of common disorders. अच्छा, अब इसमें problem ये है because we know that this is a common SNP. So if we take without disease be normal individual, so there is a difference in the allelic frequency because of course सब में एक SNP तो नहीं हो रही ना, but if our sample size is low, it's less than thousands of cases, so it's really doesn't add anything into the scientific knowledge, because mostly there are more chances that the significance that we are getting from this kind of studies is biased. So cheaper hai, and then we have the candidate gene. In that one, it's pura genome we are looking. Here we have only specific cases that we are looking. So now, what are the challenges in this genome-wide association studies? so we know that complex disorder are not caused by single gene or variant. And it's a less determining and more problematic. And we know that most of these disorders have like these SNP, if you remember the previous one, the more common variant is the rare, the lower effect it have, like phenotypically uska kam effect hota hai. So we cannot really calculate how it's going to affect the disease phenotype. And then of course we don't know how these, I mean, for example, there's SNP in one gene and then there's SNP in another gene. So how they are going to interact. So gene to gene interaction, we don't know anything about gene to environment interaction, and gene to environment correlation, and the functional interpretation. So for example, if I get a single gene mutation in an epilepsy, so I can generate that mutation in stem cell. I actually did that. We generated the mutation in stem cell. We made the neurons, a cortical interneuron. We did electrophysiology and all these functionally techniques, and at the end we concluded then yes, this is a gene that is causing epilepsy in our patient. But if I have like hundreds of 108 time loci hai, so how I'm going to functionally evaluate it? So these are like quite a lot of challenges in this kind of studies. But now what people are doing, it's called polygenic risk score. So for the SNP, those have multiple time kind of, significantly associated with this phenotype. If there are certain SNPs, those are associated with cardiovascular, a type of cardiovascular disease or hypertension in different populations. So we know that this might be something that is causing, and we somehow, if we manage to get some functional evidence. So now there are tests based on millions of common DNA variant to actually check the hidden disease risk. So for example, if I mean, there are common variant for Alzheimer and other disorders. So if I want to get myself tested, so I can get just send my DNA and get a polygenic risk score. So how many pathogenic or how many common variant I have in my genome that can lead to a common disease. But then, and what will be the benefit of this? Me sending my samples. So we check the polygenic risk score, and of course, then for example, if this is a jar and these are the genetic factors that I have, and of course there are some factors, for example, for Alzheimer's disease, they are adding with the age, or a certain disease adding with the age, and that is something out of my control, right? I have the genetic, that's something I cannot change in the whole body. And then I have aging, and then of course, we all know that aging is something that of course we cannot control in a way. But certainly there is some controllable things. For example, there is environment. So if, for example, if you don't care about anything and environment or diet, or when it comes to environment, it's how you eat, what is your lifestyle and everything. So it's like you are filling the jar, right? But these are kind of uncontrolled things. But if you control your blue things, then maybe your jar is not full, and it can protect you from the disease. So the whole point of this polygenic risk score is to protect yourself from the disease. So for example, polygenic risk scores for familial hypercholesteremia. So when you have the polygenic score, and if you have the high score, it's mean like you are at the risk, and you have the score, and plus you can actually add the environmental, you can actually lower the score by adding into changing your dietary habits and these things. So of course, how it's more like, it's not treatment, it's not treat nahi kar rahe, but we can certainly prevent, or we can prolong the onset of the disease by taking some additional environmental care. Okay, so what is the future research or complex disorder research, and what is the whole point of doing it? So one is that we have to find the genetic makeup, like the SNPs markers in genome-wide association study. And then not only the genetic, but then we also look on the epigenetic. For example, how our DNA is packed, histone modification, and what is actually, so here is chromatin accessibility, like which part of the chromatin are accessible for the transcription factor, wo kaise aake bind karenge, and all these things. And then of course, we also have to study the gene to environment interaction. For example, if you are a smoker, how that is going to change your epigenetic pattern. For example, maybe your chromatin accessibility is changed, or something is happening with, and how your genetic picture is changed by your environmental picture. Then of course, your physical activity, your exercise and all that, and then your diet and all these things can actually prevent you from the diseases. And then the other thing, the whole point is, for example, we earlier mentioned also that there are some protective variant, and then based on then risk of the disease and plus the polygenic risk score and plus the protective variant, we can actually go for the individual treatment option. And for example, according to the genetic picture, we go for the treatment options. And this is something not, I mean, of course, for the complex disorder, our progress is a little bit slower as compared to the rare disorder or the monogenic disorder. And for monogenic disorder, we are certainly going there. I mean, when we identify the genes and then we go for we identify the biomarkers, and then we go for we treat those pathways, और उसके लिए हम drugs designing and इसकी तरफ जा रहे हैं. So, do you have any question?
Chat box में question है.
Yes, I do not understand how epigenetic changes can inherit it when happen somatic cells like forms. Okay, all epigenetic changes, for example, now we know that the chromatin modeling ki changes, there are some the chromatin modification, they are in our genetic signature, and those are inherited. But of course, certainly the somatic one is not inherited. Okay, the common example that I would like to give you, for example, you know, in cancer cases, we know that there's a lot of mutation built होती हैं, but वो mutation क्यों built होती हैं? That's not only your diet or your smoking or these things. It's you are predisposed to you are predisposed to have more somatic mutation. For example, if I have a mutation, a point mutation in a gene. Actually, we found one patient. She had mutation in NF1, NF2 gene, and then she actually, because एक mutation होना is kind of, you are already at the risk. And then because आपका जो DNA repair का जो gene है उसमें mutation है, तो वो जो DNA आपका replicate हो रहा है और उसकी proof reading को वो सही तरह से नहीं कर रहा, तो it's mean you are accumulating somatic mutations. So, I mean, there is interaction, there is a link between your inherited mutation and your somatic mutation. But somatic आती भी तो सिर्फ और सिर्फ आप जो है वो environmental factors की वजह से नहीं आती. Otherwise तो हम सब में मतलब you know, the environment we are living and we are especially बहुत सारे जो लोग हैं, it's they are not smoker, but certainly they work in some industry and all these things. But why do everyone who is working in industries, or why they are not getting diseases? Because of course they are not, most of them are not predisposed. So it's like if you have the initial hit, तो फिर वो आपको predispose करता है, and then you have the chances to have somatic mutations. Okay, if anyone have question, because now we are just moving at the end, some initial जो है future directions, so where the genetics is going, so how it's evaluating, evolving. Okay. The co-occurrence, when it comes to rare diseases, so of course the monogenic have exactly the same genetic profile. Okay? Because they don't have a different genetic profile, but because they are coming from one zygote, and so they have the same. But of course, in terms of schizophrenia and other complex disorder, we really don't know the answer yet, because of course we know that the risk is not 100%, because rare mein to 100% ही होता है, but इसमें 100% नहीं है, तो it's mean there might be some other factors, either environment and other regulatory changes that we really don't understand, because that's why the heritability of complex disorder is challenging. And the dizygotic, since they are coming from two different zygotes, so there are changes. For example, if my genetic beside very common, because inheriting from the same parents, is different from my siblings, because there are some changes okay, either because of my parents have two different allele for every autosome, and then of course maybe the one that I inherited is different from my siblings. So that's why, and of course आपकी जो individual changes हैं still makes us different. So वो जो dizygotic है, it's more like a sibling, and then monozygote is exactly the same genetic makeup. But rare के case में तो so similar disease होनी चाहिए और होगी, लेकिन complex के case में नहीं होती. Why nahi hoti? The only explanation so far we have is that environment or other factors, but we really couldn't figure out those other factors yet. Okay, so this is a very famous cardiovascular geneticist in Germany. So she's basically head of a cardiovascular institute in Lübeck, Germany. So when she was 3 years old, and she had this condition, I mean, she had some muscle weakness on all these things, but later, of course, parents were normal, to kisi ko wo us tarah se couldn't identify basically. So, and then she looked later, like she herself is a genetic researcher, and then when she was in college and in university, so she had to go to because of her condition, she was not able to restore her energy, and then that's why she started living in like kind of a oxygen ventilator or ventilator tent, I think so they call it. So it's like at the night she used to put herself on the ventilator, that in the morning she have enough energy to perform her functions. So and then she found one day, I mean, she couldn't find the answer of her condition, because it was certainly not telling more about the clear clinical picture. She looked in the internet, put her symptoms there, and then she found a gene, collagen 6A myopathy. And then she did whole exome sequencing, and she found exactly a mutation. So it's like 45 years to diagnosis. Why I put here, because now there is this paper in New England Journal of Medicine, I think so it's like a couple of months ago. So what they did is they diagnosed a rare condition. So in 16 hours. So basically, वो पूरा sequencing का part उन्होंने 13 घंटे में किया. So they actually put this timeline here. So genomic and medical genetics, neonatology, personal, discuss the patient. Patient comes with emergency department, crying, or uski, I think so epilepsy thi, usko kuch epileptic seizures. And then they took the blood sample, DNA liya, and then of course they did sequencing. Then they did analysis. So it was only 13 hours, and then they actually identify jo bhi uski mutation thi. It was in biotin and thymine ordered, because of course it was biotin deficiency, and of course it was delivered to the patient, and patient was stable, and then it was discharged after 5 days. So this is where the future of medicine is, genomic medicine is. You have the patient, and I know someone personally, like one of my friends' family, so they had 5 or 7 deaths with the metabolic diseases, and it was undiagnosed. But then the couple moved to Sweden, and so the baby was born, and after 2 days the baby got the symptom, and then of course they were quite worried. But later, of course, the clinician identified the mutation, and they started giving the right dose of the diet and all these. Now he get the customized diet, and he celebrated his 3rd birthday, but 7 child in his family died with this condition. So now we know the cause, and then there are certain conditions that can be cured, that can be at least symptomatically treated. But what is for the other condition जो कि symptomatically treat नहीं हो सकते? We're going to that as well. So what is the point of this panels और जो हम epilepsy के case में और इस cases में करते हैं? So again, I put this example just to link it with the genetic finding, with the clinical, and the not only the diagnosis but the treatment. So you know, we all know that there is 70%, 30% or 1/3 of the epilepsy is drug resistant, because epilepsy में 300 से ज़्यादा gene or 500 genes reported हैं. So, but the drug that we are taking is either they are blocking sodium channel, या GABA को change कर रही हैं, या calcium को block कर रही हैं. But if the mutation is not in GABA or in a calcium or in sodium channel, so what would be the, you know, if the mutation is somewhere else and it's not gonna, you know, you cannot treat it with the same drug. So that's why we have a lot of drug resistant epilepsy. But knowledge that we get from the genetic identification, उसका certainly ये नहीं है कि हम हर एक मर्ज के लिए बीमार जो है वो नई ड्रग बनाना शुरू करेंगे. But when we have the genetic knowledge and when we have the background knowledge, then we can prescribe for the right amount of the right, not only the amount but the right type of drug. For example, if there is a mutation in this second last gene SLC2A1, so it is a glucose transporter. So अब ये GLUT1 deficiency इसको हम कहते हैं, glucose transporter 1 deficiency. तो उस केस में glucose transfer नहीं हो रहा, तो what can be the alternative? You can give the ketogenic diet, and now we know in ALDH7A1 gene, and then you can actually prescribe vitamin B6. And similarly, like you know that if there is a mutation at SCN1A, तो आप sodium blocker को avoid करते हैं, because अगर आपके SCN1A में mutation है, जो Dravet disease, very common, very one of the common epilepsies, so आपने sodium वाला उसका already block हुआ है, आप उसको मजीद block है ना, तो this is going to make the patient swear finding size medicine. So that's the whole idea of precision medicine, is we take the population, we look for the, अगर हमारे पास एक तो है कि we have the drugs and हम उसको कर सकते हैं, but certainly अगर हमें genetic पता चलती है, हमारे पास उससे alternative drug नहीं है, तो how we are going to go further. So we identify the biomarker and then हम उसकी base पे drugs बनाएंगे. But then as a clinician, you may have not noticed, but that you know, the drug that you prescribe to your patient, sometime जो recommended dose है वो आपके work नहीं करती, because of course we have a different genetic makeup than the European or American population. So last slide, you are talking about, sorry, this one?
Yes, ma'am.
Okay. Do you have question on this?
No, ma'am. I want to see this slide again.
Okay, there are a lot of this slide I took from one review article. If you want, I can send you that, and if you want any of the articles related to the things that I have explained today, I can send you. I have written my email at the start. So there are a lot more than this one that we actually can go, and not only for epilepsy, for other disorders as well. So for example for dystonia, अब एक तो वो है कि हमने ड्रग बनानी है, उसकी राइट डोज डिसाइड करनी है बेस्ड ऑन आवर पॉपुलेशन स्ट्रक्चर, but what else we can do? So आपको वही वाली go back to that manuscript वाली stage जो कि हमने कहा कि हम DNA is a human genome is a book of 46 chapter of 45,000 genes and each genes have letters. अब वो letters को हम change कर सकते हैं? तो mean you know if we have a mutation and अब वो mutation तो आ गई है DNA में, तो now we have these techniques where we can edit those. तो आपको मैं एक book दे देती हूँ, मैं कहती हूँ कि go and change the मैं आपको PDF sorry Word की file दे रही हूँ and I'm asking कि okay go and change the page 240 पे a word is you know there is an additional word so change that, or there is a word जो कि जिसमें कोई alphabet extra है या delete हुआ हुआ है, so you have to edit that. So this is like so जो हमारी normal हम manuscript editing करते हैं या वो चीजें करते हैं, this is what we can do with human genome as well. So of course the human genome editing have been one of the rapidly growing field in the biotechnology for last few decades, and especially last decade I would say, because especially there was a technique that was you know explained, it was a natural mechanism present in the bacteria, it's called CRISPR-Cas, and then the two ladies who actually worked on that one, so they described it and they got Nobel Prize in 2020. So again simple bilkul wahi wali baat hai that now you have a tool where you can change it. So how you change it and what are the difference these changes can bring. So for example there was this one news in 2018 January that the CRISPR is coming to clinic this year. So CRISPR is the technique जो कि हम gene editing करते हैं. So what they have done, they actually treated thalassemia. आपको पता है कि जो genetic disease आ गई है वो तो आपके अब DNA में हर cell में वैसे है. तो how we can treat that? So हमें पहले ये पता चल, we used to talk about the gene therapy and all these things, and now we have this one. So what was the whole point of this this study? That what they did is, so they have the thalassemia mutant gene है और उसकी वजह से कोई protein नहीं बन रही. And okay, before we go to that part, we have adult hemoglobin, और adult hemoglobin जो है वो mutant है तो उसकी वजह से वो protein नहीं बन रही. And then we know that there is a fetal hemoglobin जो कि जब adult से पहले तो हम you know that is functioning, लेकिन जब when we are adult or like after that a certain age, then we don't use our body doesn't use fetal hemoglobin, तो वो fetal hemoglobin के एक और gene आता है और वो उसे suppress कर देता है. But now you are not needed, I don't need you, so you don't have to make the protein, right? But अब जो adult ने जिसने काम करना था वो भी नहीं काम करी because there is a mutation. So they were quite intelligent to find the suppressor जिसने fetal को suppress किया हुआ था, and fetal was not available, so they removed this gene. And जब ये वाला gene remove हो गया suppressor, so now fetal is available. तो adult तो काम already नहीं कर रही, but now the fetal can function in the same way. So they did it and they published in New England Journal of Medicine. You can read this paper. It was last year in December, and they did it for thalassemia and sickle cell anemia. And they actually, it was a clinical trial for 16 months, and both the patient was transfusion independent. तो अब देखें, you cannot delete any, even ये जो आपका BCL11 है जो suppressor था जो उन्होंने delete किया, वो कोई और function भी तो कर रहा है ना, तो उन्होंने आप हर cell में से तो नहीं delete कर सकते, तो आप now the question comes, so where you will do this editing? So of course for microcephaly and जो हमारे complex neuro related disorders, and especially the one who are affecting early development, or जब when the baby is born he or she already have disease, तो उसमें हम ये नहीं कर सकते. But for example, जो blood related disorder है, leukemia है, and then thalassemia and all these things. So what people can do and what they have done is they took hematopoietic stem cell और उसमें से उन्होंने वो जीन निकाल दिया, बाकी पूरे जीनोम के अंदर जो उस जीन का फंक्शन है वो सही कर रहा है, सिर्फ उसने अब एक सब पॉपुलेशन और सेल में जो वो फंक्शन कर रहा था, जो हीमोग्लोबिन को स्प्रेस कर रहा था, वहां से हमने उसको रिमूव कर दिया. नाउ वी पुट द सेल्स बैक इनटू द पेशेंट बोन मैरो एंड नाउ द पेशेंट आर फाइन. द अदर वे ऑफ डूइंग इट, आई एम नॉट गोइंग इनटू डिटेल. I have worked with these two techniques like CRISPR and induced pluripotent stem cell as well. So for example, if you have any question in future, so you can write me and I'll be happy to discuss or happy to answer. So the other thing is, now we know that अब हमारे stem cell तो पूरी body में नहीं होते. You know, we have a limitation of stem cell, और जो होते हैं वो भी एक certain type of cells बना सकते हैं ना. For example, ये जो hematopoietic stem cell है उनकी भी एक certain lineage है. But अगर मुझे I want to study something with the neural tube defects, or I want to study something with the neural crest defect, और तो अब मुझे तो बहुत early development में जाना है और वो मैं नहीं जा सकती. One option that I have is, I can take जो IVF in vitro fertilization के embryos होते हैं उनसे cell ले लूं. But then there was a scientist in Japan. So what he did, he took our normal cell, जैसे आपके skin cell है, blood cell है, कोई भी somatic cells, उसमें उसने कुछ transcription factor add किए, and then because of those transcription factors, those cells become like stem cell. So exactly similar to the cells that are at the blastocyst stage. So now we have this ability to make any cells from this population that we call induced pluripotent stem cell. So basically, you take the somatic cells here, you reprogram, make them iPSCs. iPSCs is induced pluripotent stem cell, and then of course वो आप patient से ही लेके उसी से आप जो है वो दोबारा उसको दे रहे हैं, correct करके दे रहे हैं. तो इट्स मीन के वो आपकी जो इम्यून रिजेक्शन वाली चीजें भी है ना, वो भी नहीं होगी. सो दिस इज अ वे. सिमिलरली पीपल हैव डन सिमिलरली विद द कैंसर. सो दे टुक द नॉर्मल टी सेल्स, उसमें उन्होंने क्रिस्पर से कुछ जो इसके नॉकआउट किए थे, PD1 नॉकआउट एंड एंडोजेनियस TCR नॉकआउट. सो वो करके they gave back to the patient and the patient survived. It was some paper in the science. So now we have this thing that we can do genome editing, and we can actually correct the genetic disease, genetic mutation. But again, we are limited, but there are trial going on certain disorder. For example, ALS में आप करते हैं, आप iPSCs लेते हैं, patient से बनाते हैं और आप उससे neurons बना के, और but again there is a lot of research needed, and we have to do a lot. Blood brain barrier को कैसे cross करेंगे and all these things, but at least it gives us some direction that where we have to move. Similarly, there's again another CRISPR variant where prokarya जो है early aging उसको करने के लिए, this was again recently in January 6. So this is a group in Harvard. So they are doing not only for this disease, but also for sickle cell anemia and muscular dystrophy. And there are a lot of groups in the world who are doing similar kind of work. So this is our second last slide. I'm telling you that you, because of the time and you don't get bored. So now we have these all wonderful techniques, genomics, editing, stem cells, सब कुछ है. But then where we have to change. For example, अब वो जो spectrum में हमने बताया, जब तक हमें पता नहीं है कि ये mutation, ये disease कर रही है, तो हम उसको जाके तो नहीं correct कर सकते. And similarly, if there is a mutation that if the person is fine or वो polydactyly है, तो not impact on a huge impact on the human health. So now you have to we have to think about like what is needed and what background knowledge we have. For example, अब आज मैं कहूँ कि okay, I'm going to make this stem cell and I'm going to treat Alzheimer's disease. No, I don't know how to, I mean, what we are giving to the brain patient, maybe it is going to, agar usme koi aur population cell ki aa jati hai, तो it can certainly make a disaster or make the patient phenotype even severe. तो आपको the background knowledge, the right research question और ये सारी चीजें जरूरी है. So this is the example जो कि first genetic edited babies थे. I'm sure you might have seen this. So there was this guy from China, so he wanted to be kind of the first person to make genetic edits. So what he did IVF, in vitro fertilization, और उसमें ना एक जीन उसने CCR5 जो कि एड्स की ससेप्टेबिलिटी को इंक्रीज करता है उससे डिलीट कर दिया ना, कि अब ये बच्चों के फादर को तो शायद ये नहीं कि ये अब इनको एड्स नहीं होगा. And then of course he implanted the edited embryos and then the 2 twins girls were born. अच्छा, अब जो सबसे बड़ा इशू है, this CCR5 was reported from I think so from European population, but the Chinese population is different. For example, अगर अब American population या South African पे कोई काम हुआ है, तो I need to first look in my population, what is the structure of my population, what is the status of this polymorphism, or what is the status of this mutation. Maybe ये मेरे वाले population में normal हो. And then based on those knowledge and things, then I decide. And or the other the biggest problem with this genome editing thing is that it's kind of for example, अगर मैं आपसे कहती हूँ कि okay, go and 220 पेज पे जो है वो word genetics को आप change कर दें, या transcriptomics को आपने proteomics करना है, you go and अब वो transcriptomic का words उस 220 पेज के अलावा भी बहुत सारी जगहों पे है. So you go and control F and then you find transcriptomics, go for the genomics or whatever. So it's called off target. तो आप जो भी enzyme और ये चीजें दे रहे हैं सेल को प्रोवाइड कर रहे हैं, तो it can be somewhere else and that can actually change, make some change that is more lethal. So we really need to have the right question and thoughtful strategy to answer that question. So yeah, so genetic change your genetic destiny. So of course, if you have the polygenic risk score, तो उसको भी हम कर सकते हैं. Now we know that we can edit the genome and we can all do all these wonderful things. So yeah, so this is all about this one. So please ask question if you have. So the last part was because I just wanted that you all are doctors and you are going into the clinical diagnosis, and that what are the future of this whole genomic medicine, where are the possibilities of all these where we can use these all wonderful techniques. So anyone have question? Okay, someone. Thank you.
मैम, ये जो आपने ये वाली स्लाइड लगाई है, चेंज योर जेनेटिक डेस्टिनी बाय डॉक्टर जो डिस्पेंजा, तो मैम आई हैपन टू हैव read some of his research. मैम, he says कि आप अपने थॉट से अपने जेनेटिक कोड्स को बदल सकते हैं और ये वो, तो मैम क्या is it possible, like if he saying the right thing?
No, I don't agree. I just put this slide just to at the end वो वाली उसको करने के लिए just for example the polygenic risk score पे हमारा lecture था, तो उस वजह से. But definitely I don't believe that the thought all this, okay, हमारा जितना भी thought process है and everything is normal, because I cannot be rude, I cannot be respectful. So and मतलब हमारे जो वो filters हैं वो तो लगे हुए हैं ना. And for example, if we lose those filters and we have some behavioral problems and those things. So genetics certainly cause those things, and हमारी जो behavioral changes हैं और जो neuropsychiatric disorder है, for example, we know that in kids we have autism, and autism have 90% genetic heritability. तो वो वाली चीजें तो हैं that we filter the noise, we filter the other signals, but I don't think so. And I'm sorry, I haven't read any of his like that kind of a thing. So I might mean, अगर मैंने पढ़ा होता तो maybe I would have like a better answer. But this was just to say like that, you know, genetics is important, and certainly कुछ चीजों में तो सिर्फ genetics ही important है. But then